My name is Max Cantor, and from my biology role model project, I will be talking about David Liu. David Liu was born in Riverside, California to Taiwanese immigrant parents. He was always interested in science growing up, but he credits a lot of his interests to his father, who was an aerospace engineer, and his mom, who was a physics professor. He was always outstanding academically and graduated first in his class at Harvard University in 1994. He would go on to receive his PhD at UC Berkeley in 1999. Afterward, he was immediately offered a teaching job at Harvard University and eventually became a full-time professor at Harvard by 2005. Later, he would even join the he would become a Jason Academic Science Advisor in the US government in 2009. Although he is busy doing research, Dr. Liu does enjoy a number of hobbies, including woodturning, photography, and video games. He also enjoys gambling, which he claims is a great use of applied math and statistics skills. Dr. Liu's research focuses on genetic engineering and editing the genome. His main goal is to harness the power of evolution to solve problems within the human genome. His current research focuses on DNA templated synthesis, protein evolution and delivery, and genome editing. He wants to find out how we can use genetic modification and evolution to cure genetic diseases. Dr. Liu has published over 180 research papers and is the inventor to over 70 patents. His most recent paper is called The Evolution and Minimization of Cas9 Independent Off-Target DNA Editing by Cytosine Base Editors. Essentially, the objective of his experiment was to identify and engineer cytosine base editors that minimize the need for Cas9 and DNA editing. The methods he used to do this were to combine different variants of cytosine base editors and basically put them into E. coli. The E. coli were then exposed to rifampin, which is an antibiotic that kills bacteria, and the survival of E. coli was measured. And eventually he determined that the E. coli that were given these cytosine base editor variants were able to survive better in the rifampin, showing that these cytosine base editors are a more efficient and cost-effective way to edit the genome. In addition to his research, Dr. Liu has a number of other accomplishments. He is the founder of 7 biotechnology and therapeutics companies, 4 of which are listed to the right of the text. He has received a number of awards from Harvard for his teaching and a number of awards for the research that he has done in biochemistry. In 2016, he was named one of the top 20 translational researchers in the world. In 2017, he was named one of Nature's top 10 researchers. In 2020, he has already been granted the ACS Chemical Biology Lectureship Award. I identify with Dr. Liu for a number of reasons. First, he was born in Riverside, California, which is about an hour away from where I was born. He is also an Asian American, just like me. I admire the way that he took control of his own career and followed his own interests and was able to make his own career out of it. He also worked extremely hard on all of his research and all of the projects that he's ever done. And finally, he branched out. He didn't just stay in Riverside, California his whole life. He went to Harvard, he went to Berkeley, and he stepped outside of his comfort zone. He took risks and they ended up paying off for him, and I think that's what I admire about him most. Dr. Liu has made a number of discoveries. Some of his most important include prime editing, base editing, phage-assisted continuous evolution, and DNA templated synthesis, all of which are different ways of increasing the rate of evolution and editing the genome. He has been pioneered as one of the world leaders in the CRISPR 2.0 revolution. His most important goal right now is to use his genetic modification expertise in curing a genetic disease in humans.